Okay, everybody, let's do some speciation. Okay, that's what these notes are called. They're going to be about species and how changes in a population lead to the production of a new species. So, speciation was addressed in Darwin's book, um, and a lot of questions were coming about, like how did living organisms or species get to Earth, and they were just so confused back then. Um, about how things really started and why are there different things living now than there were in the past. All that's really confusing until more time passed and we learned how to figure out more things. But Darwin, despite the title, didn't address the origin of species hardly at all, because they just didn't know at the time. So it's kind of a peculiar title. Um, the book itself really deals with the process of natural selection and what he called descent with modification. That was his first name for his kind of theory that he was working on, was descent with modification. So if we look at some species concepts, there's actually more than one. So there's the biological species concept, defined by Ernst Mayr. And if you go into anything biological or especially environmental or ecological in college, you will learn about Ernst Mayr. He was one of the first kind of big time ecologists. And so he defined a species as a group of individuals whose members have the potential to interbreed with one another in nature and produce viable fertile offspring. So basically, you have to live around them, you have to breed with them, and your offspring have to be able to breed. Okay, so that's the biological species concept. And so here we have an example of the northern leopard frog and the southern leopard frog, virtually the exact same frog, but they do not live near each other, so they can't breed together. So they are separate species according to this model. Then we have the phylogenetic species concept, and this was grouping things or species based on their physical appearance, their characteristics. It's really the only thing we had forever to group species together was what do they look like? Are their structures the same? Is the bone structure the same? All that sort of thing. Benefits is you can apply this to anybody. Drawbacks is appearances can be deceiving, like all things with wings are not birds. Okay, so it can be tricky on how to place things just based on appearances. There's the evolutionary or paleological species concept, which is based on fossils. Again, you can apply it to everything. Drawbacks, it's hard to connect because it's hard to connect the lineage because not everything leaves a fossil. Not everything makes a fossil when it dies. So a lot of gaps that way. Um, the ecological species concept, then, as you might suspect, being in ecology, is based on niche and all the little roles that organisms play, and only one thing can occupy one niche. So that would be its own species. For every little niche, there would be a particular species. And then, um, if the same species is always going to compete for the same resources, and so new species evolve from this competition. And we're going to delve much more into this when we get to ecology later in the spring. So why do we need all these definitions for a species? It's really as simple as it depends on your angle. It depends on what type of discipline of biology you're in and what you're researching and how you want to go about things. But there's all these different ways to do that. So speciation for our purposes will equal reproductive isolation. So when species get isolated and they reproduce together, then you're going to be deemed a species. Okay, so they result from a lot of series of evolutionary processes that create this isolation, this reproductive isolation. So lots of processes can do this. Okay, so there's, oops, my mistake. There is anagenesis and cladogenesis. So anagenesis is where a single species transforms into a different species over the course of several generations, and you can see this at the bottom. And remember, it says over several generations. Okay, so it's not that one thing turns into another automatically in one generation. That arrow there is representing several generations. And then cladogenesis involves the division of a species into two or more. So if you look at the one on the right, The original bird, the original finch down there, the big gray one at the bottom, is still there in the top picture where there's three of them. The original is still there. You just have other variations now. Where if you go back to anagenesis, the original would be gone. It would have transformed into something else, and the original is probably gone. Allopatric speciation, or what we call other country. That's literally what it means, is other country. You don't have to take country so literally. It just means like other space. Um, so, if you look at the little diagram to the right, it's how you might kind of envision something like this going on. So you have an original population. Then, for whatever reason, the population gets separated, and there's lots of reasons for that that you may think of. And then, one of those population changes because of the pressures they're under is now different. And so now we have a little difference in the population. And then, you have these new distinct species, and maybe they overlap, maybe they don't, just kind of depends. Um, but lots of barriers can cause um, these separations. For one, maybe an isthmus. Okay, the Isthmus of Panama. Okay, when it arose about 3.5 million years ago in the Caribbean. So that lent itself to having several different types of species, whether it be fish or something else, on one side of the isthmus versus the other. Sympatric, or same country, same area, same space. Populations occupy the same physical space and yet become reproductively isolated. What would possibly do that? Look at this picture over here. You have this green circle, then you have this yellow color showing up within the green circle there, within the green population. There's no barrier, there's no other pressures, they're under the same pressure. So what would actually cause that? And you can see the word genetic there, so maybe that's a clue for you. Something like mutation might cause something like that in a group, and then it becomes maybe a little advantage, and then you end up with these two distinct species. So just a little summary slide, allopatric versus sympatric. So the original population up top. On allopatric, remember that's other country, they get divided by space, so the water level drops. Now the populations are separated, and you end up with some blue ones on the left and some orange ones on the right. Where sympatric, they are in the same space still, they can mingle and breed, and the blue probably came about by mutation. Isolation effects. So isolation does not always lead to speciation, okay? So a lot of times you might have hybrids, and hybrids aren't exactly their own species. They're kind of these, links between related organisms, let's say. And this is typical of what they call ring species. So if you look at the top of the globe, a lot of the continents are actually fairly close to one another, and organisms actually kind of traverse a lot of that around the ring, around the Arctic Circle up there. And so if an organism starts out at like number one, okay, and then they can mate with number two, and then two can mate with three or one, and then three with four, four with five, five with six, six with seven. But then when you get around to seven, notice there's nothing between seven and one, and that's because all the little changes around the ring have gotten to be so different that seven and one are not really, they're not able to mate, or they don't want to mate, or they're just not compatible to do that anymore. So hybrids can kind of show up this way. So here's a little bit about, hybrids and some possible outcomes. So the only way that you're going to get to new species is if that hybrid zone disappears. Okay, so probably need my pen for this one. So here's the original population right here. Okay, so this group right here is your original, and it says gene flow, right? So we're mixing of genes, right? But now this red little hook right here, and it says barrier to gene flow. So in this case, this group is going to go off in this direction, okay? And all of this group is going to go off in this direction. And these guys are still having gene flow go on, and these guys are off doing their own thing now, okay? Because there is this hybrid zone, this area in between. And so in this case, they can get back and forth to each other. You see the arrows on each side, and they can get back and forth. Okay, so there's a few possible outcomes here. Okay, So, and here are the three words. We have reinforcement. I'm not drawing straight lines tonight. We have fusion, a little better, and we have stability. Okay. So in reinforcement, you lose touch. Each side, okay, this arrow and this arrow loses touch with the hybrid zone. So now you have these possible outcomes. So reinforcement would be they actually become their own thing. They're reinforced and they're going to become their own thing. Fusion, they were separated, right? We have this going on, but then they come back together. Sorry about the terrible drawing. Um, Move my mouse up on this folder. Maybe I'll have better luck. Um, but right here, if you look, the hybrid zone is right there in that little area, and now they've merged back together, and now they're back into just this one guy right here. But you might have had some characteristics from both of the sets that then merged back in, so they're fused, right? You get some new options in there, even though there's only one thing, one organism now. And then you get stability, which is basically we're just going to keep existing like this. The hybrid zone is just going to keep on keeping on. And so you get, you have some of these guys up here, and you have some of the hybrids in the middle, and then you have some of these guys down here. And you're going to have these different variations of it, and it's just going to stay that way. We'll go through some examples like that later. So reproductive barriers. There are also various barriers to reproduction that prevent successful reproduction. And so remember what successful means, right? Just surviving long enough to pass on your traits. Okay. So there are many barriers, and the way we distinguish these barriers is by time, by when they happen. Do they happen pre-zygote, okay, pre-zygotic, or do they happen post-zygote, post-zygotic? If you don't remember what a zygote is, it's when the egg fertilizes the sperm and you have that first cell in reproduction. So is the barrier prior to the actual reproduction, or is it after? Okay. I guess I should have said fertilization since that's what the slide says. All right, pre-zygotic, before fertilization, habitat isolation. Maybe they just live in different areas, okay? Kind of like the frogs earlier, where they just aren't near each other, right? So that's habitat isolation. They're not near each other, so they're not going to mate. That's a barrier. Another example of the habitat, or I'm sorry, a little tired. This one is called temporal or temporal. If you're not sure what the word temporal means, it actually has to do with time. Okay, temporal always has to do with time. So in this case, the species breed maybe different time of day, different season, different year. You have the spring cricket down here, and then you have the fall cricket. And again, virtually the same cricket, but they don't breed at the same time. One mates in the spring and one mates in the fall. So that's a barrier to reproduction. Oh, these guys are pretty funny. So these are the blue-footed boobies. Um, they are birds that are actually on the Galapagos Islands. And these guys have what's called behavioral isolation. So this happens when organisms have these, distinct mating rituals and patterns and dances and calls and things like that that they're trying to do to impress a mate. We actually watch a video about these guys in class. I will send out the link in an email or something so you guys can watch it. It's pretty hilarious to watch these guys strut their stuff. That's the comment about the video, because they're blue-footed boobies. Another one is mechanical isolation. Reproductive anatomy is not a match. Unfortunately for these snails, they just can't make it work. Their anatomy does not go in the same direction or in the direction that it needs to in order for mating to happen. So they want to mate, they just can't physically make it work. So that is a mechanical isolation. And then what I believe is our last pre-zygotic barrier here is gametic isolation. So they actually mate, they try to reproduce an organism. There's actual sperm and egg there, but the gametes, the actual sperm and egg won't fuse. The proteins on the surface of the membranes just don't jive, and so they're not able to fuse together, and thus they can't form a fertilized egg. And so that's gametic isolation. Then we move to post-zygotic barriers. So as you can see, we have an offspring. So these are all things that happen after the offspring is born, thus post-zygotic. The first one is reduced hybrid viability. So maybe two things mated that weren't the same species, and they created an organism, but the hybrid is weak and it generally doesn't do very well. And this guy's kind of frail, and he just probably isn't going to make it survive very long. And as a species or as a group, these hybrids don't make it very long to become their own group. Then you have reduced hybrid fertility. So in this case, um, you have mating going on, you have the organism, seems healthy, okay? But in this case, the one at the bottom down here, which is the mule, okay, the mule is sterile. And the mule comes from a male donkey and a female horse. And so um, the mule's a perfectly good animal, really good farm animal, hard worker, but he can't reproduce. And so you have this reduced hybrid fertility where you can't just have, mules all the time mating with other mules. They only come from donkeys and horses. Hybrid breakdown. Um, this is where the fertility of the hybrids gets reduced over generations. You see the big tall plant in the front, and then a few more generations go by, and the plant gets smaller, and then you can see in the way in the back of the little guy, and he's not doing so well. So over time, the hybrid just keeps kind of getting more and more frail or more and more weak, and just isn't very viable over the long haul. And that's it for post-zygotic, just a few of those, many more pre-zygotic barriers. So a lot of studies people do, and we've seen it in your misconceptions where they say, you can't see species, you can't observe evolution, and we know that this is wrong. We've had plenty of examples about why this is wrong. So I thought I'd give you some more reminders about um, studies that are contribute to the speciation and or not, because a lot of these are barriers. So I'm going to throw up some slides, and they're just examples. And rather than me say this every time, what you should do is when I throw up the slide, I'm going to give you a quick pause, and you should pause the video and see if you can think of whatever in the slideshow that I've shown you so far, which word or term or concept applies to each slide. Okay, so pause each one and then try to come up with what it might be, and then I will obviously reveal the answer so that you guys know what's going on. All right, so in this case, two different species of antelope squirrel, one for each rim of the Grand Canyon. So um, take a shot. This one should be habitat isolation, right? Because they're not near each other. They have a giant canyon in between them, so they're not going to mate. So pre-zygotic barrier. In this case, you have the formation of the isthmus. And while we did apply another term to this, I want you to pick a pre or post-zygotic barrier. It also fits the other term you're thinking of, but go ahead and pick the pre or post-zygotic barrier. And again, that would be habitat, right? Because you're split, okay? You're not near the other group to mate with. In this case, a lot going on in this slide. So pause it and really, really try to understand this, because stuff like this shows up all the time on like an AP exam where they're making you analyze stuff like this. So at the bottom, genetic analysis of the toad population of Europe shows there is only a narrow zone where hybrids between the yellow-bellied and the fire-bellied toads can be found. So if you look at that red line going right through the yellow and kind of orangey part of Europe, we're talking about that red line. You can see the blown up picture right in the middle of the hybrid zone. So take a second. And that would be called the stable hybrid zone, where it remains, right? Where all three options are remaining, and that hybrid zone stays put. Here we have the hybrid growler bear, right? The cross between the polar and the grizzly. And if you look at that bottom picture of the growler compared to the top two, the growler doesn't look so hot, and they've not shown to, as of yet, survive that well. So that would be hybrid breakdown, because they're not surviving that well. But this is happening more and more, right? As polar bears, the sea ice goes away and the polar bears are venturing more and more south, and they run into grizzlies. We're seeing more and more growers. So maybe at some point they'll become more robust and be able to make it, but it's just kind of sad that that even has to happen. And the polar bears are, in that much distress. So in this case, we have a couple populations of mice on different sides of the island, and it says they demonstrated different chromosomal fusion mutations. And there's not a lot to go on with this one, so I'll just kind of tell you that this one is really just reinforcement because you have these two distinct populations now where you had these chromosomal mutations, but now you have the red group and you have the blue group, and they are reinforced as being their own group now. With the wheat here, modern bread wheat is a hybrid species with genetic contributions from 3 wild wheatgrass. So if you look at the top, you have AA, and then you have BB, and then you have DD, and then you have the one down front that has all 3. So take a second, think about what that one might be, pause the video. So with the wheat down front that has all 3, right, this would be the fusion. This would be the fusion example where now it's taken kind of the best parts from all the hybrids and put them back into one. This one's definitely a little tricky. Okay, so experiments with transgenic monkey flowers show different pollinator preference for species-specific flower colors. So think about this one for a minute. Pause it and think about this one for a minute. So hopefully you thought about that. In this case, it's about the pollinator showing preference for a particular color. So that would actually be behavioral isolation. That would be the one like the blue-footed booby, where it has to do with some sort of behavior of the organism and whether they're going to be able to make mating happen or not. And in this case, they're basing it on color, but it's really on the behavior of the pollinator. And again, kind of a lot going on with this one. So we have we have sympatric populations of 2 flycatcher species. Remember, sympatric is same country, okay, same space, and they're much less likely to interbreed than the allopatric ones, right? So take a look and look at the left where it says female mate choice, and they both say female mate choice, and look at the 2 columns there, own species, other species, own species, other species. Why is it doing that? Why is there 2 bars on the right and 1 bar on the left? And what's really going on here? And I can actually tell you should come up with 2. There should be 2 of the concepts here that apply. So go ahead and pause the video and take a shot at that. So in this case, you're looking at a little bit of habitat, right? Because you have the allopatric populations, and so I'm sorry, the sympatric and the allopatric, like one versus the other. And so you have sympatric on the left and allopatric on the right. So it's a little bit about habitat isolation on the one side, and then it's also behavioral, right? Because whether they want to mate with the one species versus the other. So kind of a couple things going on in this. So in this case, you have a mate choice preference in populations of mosquito fish, and they're correlated to similarities in body shape between male and female. Body shape is correlated to predation pressure. So in this case, you have a lot of mating going on with similar body shapes. And so, and right off the bat, it tells you it's mate choice preference. So that should tell you it's behavioral, right? This is a mate choice preference. So this is a behavioral situation. This one, big diagram, lots going on. Pause the video, look at, try and see if you can really figure out what's going on here between all these fruit flies, and has something to do with starch and maltose, and have a bunch of generations going, and then you have male and female and that sort of thing. So see if you can tell what's going on. In this case, it would be selection based on food preference, right? So if you look, especially at the left with the female, you can see the 22 and the 20. You have the male starch and the female starch are both have a lot of matings there, and then the maltoses match up, the starch matches up, and the same thing. The other side, not so much, right, in the control, because that's just the control part. But on the left-hand side, the experimental group, it really shows up that flies that were like raised on starch, like to mate with flies that were raised on starch, and vice versa with the maltose. So behavioral again. In this case, you have this speciation study, so hybridization. So hybridization between two species of sunflowers has produced a new species in the wild. And so um, this hybridization has also been seen in the lab, and they show you the chromosome diagrams over here. Look at the ones in the background, look at the ones in the foreground. And the one in the foreground is obviously doing better, right? And they said it was a hybridization between two species. And so we actually call this one hybrid vigor. This is like a hybrid that's doing really well. Um, so kind of a fusion as well. Fusion or hybrid vigor, either one would work. Okay, last couple slides here, last couple concepts. So the rate of speciation. How fast does it happen? There's an ongoing debate. First you have gradualism, right? And this is like something like Darwin and Lyell would talk about. Remember, Lyell was uniformitarianism with all the small changes of the Earth because he was the geologist. So does it happen like this, where you have this butterfly at the far left, and there's little bitty changes over long periods of time, and by the time you get over many, many generations and hundreds of thousands of years, you have different looking butterflies. Is it gradual like that? Or does it look more like punctuated equilibrium, where the rate is not constant? There's like bursts of change, and they're followed by long periods of little or no change. And so more recent evolutionists or evolutionary scientists kind of propose this one. And so down here, like maybe the change happens this fast. Like if you look down at the diagram, you have the butterfly and they change immediately. There are mutations or something like that, and the changes happen very fast. And there's long periods of time where they don't change, and then the change happens fast. And there's long periods of time where they don't change. So do you think how do you think it works? Do you think it's gradualism, or do you think it's punctuated equilibrium, or um, maybe, just maybe, it's a kind of trick question, because the real thing is that they both happen. And it just, different parts of history, we've seen both of them. We've seen gradualism and we've seen punctuated equilibrium, and so they both can certainly occur. And then last concept here, evo-devo, is evolution and development. There's a ton of research going on in evolution and development. And so, how do things develop, and how do the genes dictate, what might develop and what things are going to look like and how they're going to function, and how do these genetics apply to the evolution of things? Okay, so it's a huge field right now. Lots of research going on. So the genes that get studied quite a bit are called Hox genes or homeobox genes. And so these are the genes that control development. And if you look at a lot of different organisms, our body plans are quite similar. Okay, they're responsible for the development of specific sections of organisms, is what the Hox genes do. Okay, and changes in expression of these genes lead to big changes. Okay, so let's look at first set here. So if we check this out up here. Okay, so we have Hox gene 6, Hox gene 7, and Hox gene 8, right? And we're looking at these. Well, in this case, this UBX1, okay, so this one is going to get expressed differently between the fruit fly and between this crustacean over here. And this segment is going to control the appendages, the number of appendages that come out of like the abdomen area or the midsection. And so insects get their 6, and then crustaceans have quite a few more. And so it gets overexpressed, so it makes more limbs in that area. And if we follow that down here to a snake and a chicken, okay, this is a chicken embryo. And if you look real closely in here, you can see like the front wing and the hind legs in here, okay? And so the expression of a specific Hox gene is purple, and it's responsible for limbless segments and chordates. So look how much purple is here in the chicken. Limbless segments. So between the front two and the back two, the section is purple because there's no other limbs in there. Well, look at this same Hox gene in a snake. Look at all the purple. It's everywhere, right? So 0 limbs. They don't get any limbs because this particular Hox gene doesn't get any expression in a snake, where other things get much more expression and we get lots of limbs. So couple questions here for you, but real quick, going back to the idea here, um, when you think about whether it's a human or a dog or a worm or a bird or whatever, when you think about the Hox genes and how they organize things, Hox genes are really well conserved over the whole animal kingdom, meaning they haven't changed much. They haven't evolved much because, the head is still where the head goes. The head's still in the front. Then you have the abdomen, you have some appendages, you have the tail end, you have the digestive tube, right? Our digestion is very similar to an earthworm, and many other organisms. And so all of these Hox genes and the way things are kind of organized, the body plan, like your biology is not that different from a pig or a dog, as far as like where your organs are and how they work. Like the body plan really isn't that different. And so um, that's what Hox genes do. They're kind of the ones that are all developed and or are all in control of the development. And there's just, little bitty changes in the gene expression that make all of these different things um, look a little bit different and function a little bit different. But all in all, the body plan's not all that dissimilar from everything else. So anyway, there's some questions for you, and I'll post these notes so you can check out just the slideshow as well. Okay. All righty. Hope that's helpful for you.